Welcome. Today, we're diving into elemental impurity analysis, guided by the expertise of Dr. S.K. Singh. We'll explore how to apply the ICH Q3D guidelines using ICP technology. This sets the stage for understanding how to ensure safety and compliance in pharmaceutical analysis. Let's get started. Our goal here is clear: to keep elemental impurities well below toxic limits throughout a product's entire life cycle. Why? Because these impurities can cause serious risks, toxicity, organ damage, even cancer or neurotoxicity. They threaten product safety. So what exactly are elemental impurities? They're tiny traces of metals that can sneak into drug substances, excipients, or finished products. They come from many sources: raw materials, catalysts, equipment, water, packaging, and even the environment around us. Understanding this helps us manage those risks better. Let's take a closer look at the regulatory landscape guiding elemental impurities. We're talking about key references like ICH Q3D, USB chapters 232 and 233, and the European Pharmacopoeia. These set the rules for drug substances, products, and excipients, but not for biologicals, herbal products unless local rules say so, or radiopharmaceuticals. The core ideas here are a risk-based approach, setting permitted daily exposures, considering different routes of administration, and managing these limits throughout the product's life cycle. Let's break down how we classify elements based on their toxicity and how we evaluate them. Class 3 elements have low toxicity when taken orally, things like copper, lithium, and tin. We only look at these when they're intentionally added. Then, Class 2B elements including silver, gold, and selenium are more likely to show up, so we check them regularly. Class 2A elements like arsenic and lead are a different story. They always need a full evaluation for every product. This system helps us focus where it matters most. When we talk about regulatory importance, it's all about meeting strict safety standards. Results have to show compliance with specific PDE limits. That's permitted daily exposure. PDE tells us the maximum amount of a substance someone can be exposed to daily without significant health risks. For example, arsenic's limit is 15 micrograms per day, cadmium and lead are both set at 5, and mercury is allowed up to 30. These numbers guide us to keep products safe and compliant. Let's walk through a practical example using a palladium catalyst in synthesis, which carries a high risk level. Because of this, routine monitoring with ICP is essential. The risk assessment follows the ICH Q3D process. First, we identify potential contamination sources, raw materials, water, equipment, and processing aids. Next, we evaluate how likely contamination is. Then, we perform analytical testing, compare results against permitted daily exposure limits, and finally, establish a control strategy to keep everything safe and compliant. Let's talk about detection capability, specifically, how we measure substances at incredibly low levels, from parts per billion down to parts per trillion. One powerful tool for this is inductively coupled plasma mass spectrometry, or ICP. It works by turning a sample into an ionized plasma at extremely high temperatures, then separating those ions by mass for detection. The advantages: it can analyze multiple elements at once, detect ultra-trace amounts, and deliver results quickly with high sensitivity across a wide range. Let's take a closer look at the key parts of an ICP instrument. Each component plays a crucial role in turning a sample into precise, measurable data. From the plasma source that ionizes the sample, to the mass spectrometer that separates ions by their mass-to-charge ratio, every piece works together seamlessly. Understanding these components helps us appreciate how this technology delivers such accurate and sensitive elemental analysis. When it comes to preparing samples, there are some common challenges we face. One widely preferred method, especially for regulatory purposes, is microwave digestion. This technique uses a combination of acids, typically nitric acid, hydrochloric acid, and hydrogen peroxide, to break down the sample. A typical procedure might start with half a gram of the sample, add 8 milliliters of nitric acid, 2 milliliters of hydrogen peroxide, then apply microwave digestion. After that, The mixture is diluted to 50 milliliters and analyzed using ICP. Let's walk through a calculation example to find a sample concentration. This will help us see how the theory works in practice. By breaking down the steps, we'll understand how to apply the formulas and interpret the results. It's one thing to know the method, but seeing it in action makes it stick. So, let's dive into the numbers and get a clear picture of the process. Now, let's look at a compliance assessment example. This step helps us see how well we're meeting the required standards. It's not just about ticking boxes, it's about understanding where we stand, identifying gaps, and making sure we're on the right track. By reviewing real cases, we get practical insights that guide our next moves and keep us aligned with expectations. Method validation is a crucial step to ensure our testing meets the highest standards. According to USB Chapter 233, this process confirms that our methods are reliable, accurate, and precise. It's about proving our approach consistently delivers trustworthy results. By following these guidelines, we build confidence in our data, and ultimately, in the safety and quality of the products we work with. When we talk about contamination sources in ICP analysis, three main culprits come to mind: reagents, glassware, and the laboratory environment itself. Each can introduce unwanted elements that skew our results. To tackle this, we rely on several mitigation strategies. Collision cell technology helps reduce interferences, internal standards keep our measurements accurate, and using high-purity reagents minimizes contamination from the start. Despite these tools, challenges remain. It's a constant balance between precision and the limits of our methods. Let's dive into a case study focused on residual palladium in active pharmaceutical ingredients, or APIs. This example shows how even tiny amounts of metals left over from manufacturing can impact product quality and safety. We'll explore the challenges faced, the methods used to detect and reduce palladium, and what this means for ensuring medicines meet strict regulatory standards. It's a real-world look at precision and care in pharmaceutical production. ICP has become the go-to technique for testing elemental impurities in pharmaceuticals. It helps manufacturers meet strict global standards like ICH Q3D and USB, all while keeping patient safety front and center. The ICH Q3D guidelines focus on risk-based assessments, patient safety, and managing impurities throughout a product's life cycle. ICP delivers powerful multi-element analysis with ultra-trace sensitivity and accuracy, supporting regulatory compliance. But success depends on more than just the instrument. It requires thorough risk assessment, careful sample prep, validated methods, and ongoing monitoring. Thanks for joining me today. If you want to explore this topic further or have any questions, please don't hesitate to reach out. I appreciate your time and attention, and I look forward to continuing the conversation. Thank you.